Welcome to Microlab Mastery. Today's topic, simple stains. Most cells are transparent. Staining them makes it easier to see and distinguish. In other words, it enhances the contrast. With simple staining, only one stain is used. This can be used to determine cell shape and arrangement. In contrast, with differential stain, more than one stain is used. This can be used to determine differences between different bacterial species. For example, cell wall structures, endospores, capsules, flagella, etc. Before staining, you must prepare the bacterial smear and fix it to the slide. To create the bacterial smear, cells are going to be applied to the microscope slide. Your starting material could be a liquid culture, in which case a loop full of culture is spread onto the surface of the slide. Or it could be a solid culture, for example a slant tube or a petri plate, in which case a drop of distilled water is placed on the slide and a small amount of growth is aseptically collected using a loop. The loop is then used to spread the water across the slide to create a smear. The smear is then dried completely before fixation. During fixation, cells are attached to the slide. This also kills microorganisms, stopping their movement and metabolism. It preserves the integrity of cellular components, and it is achieved by heating or chemical treatment. In order to perform the streak plate method of isolation, you will need the following materials: bacteria cultures, clean microscope slides, a set of simple stains, a staining tray, bibulous paper, also known as blotting paper, a slide holder, a squirt bottle filled with water, a microscope, plus lens cleaner, lens paper, and immersion oil, and materials for aseptic technique, for example, inoculating loop and Bunsen burner. In order to prepare the bacterial smear and perform the heat fixing, please follow the following steps. First, label your slides using a Sharpie. Next, please follow aseptic technique to sterilize your inoculating loop, as you have previously learned in the class and as is being demonstrated here. Next, please pick up a small droplet of water using your inoculating loop and place that droplet of water on a clean slide. If you are starting from a broth culture, then please skip this step. Follow aseptic technique to sterilize your wire loop and ensure the loop is cool before you place into your bacteria culture. Aseptically transfer bacteria from the culture to the water on the slide, and ensure you're following aseptic technique by flaming the mouth of the test tube. Ensure that you thoroughly mix the bacteria with the water and spread the droplet out. Then re-sterilize your wire loop. Please repeat the previous steps for any additional slides you are making. Next, please allow your slide to air dry.
5 minutes later.
If prepared and dried correctly, the smear should be slightly cloudy. Using a slide holder, pass the smear through the upper part of a flame 2 or 3 times. This heat fixes the preparation. Avoid overheating the slide and allow the slide to cool. Now that you have prepared your bacterial smear and heat fixed your slide, we're going to describe the simple staining procedure. First, cover your smear with the appropriate stain for the designated time. Please note the timing is critical. Follow the exact time listed in your lab procedure handout. Make sure you're using a staining tray to catch the excess stain. Next, please take the slide and thoroughly rinse the slide with water. Next, please gently blot dry your slides using bibulous paper. Ensure that you are gently blotting and not rubbing the slides with the bibulous paper. After the slides have been made, when you're viewing them with your naked eyes, they should look something like this. Your next step will be to examine your slides using a microscope. If they've been properly prepared, they should look something similar to these images. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.